Hi, I'm Techjip Ha. I'm a professor of biophysics and biomedical engineering at Johns Hopkins University. I'm also an investigator with the Howard Hughes Medical Institute. This is the second of a three-talk series where I discuss how single-molecule technologies are changing the way we study and understand many of the important biological molecules. In this part, I will discuss how the combination of single-molecule fluorescence technologies and single-molecule mechanical manipulation technologies can be used to study the dynamics of the nucleosome, which is the compaction unit of our own genome inside our cells. We have been interested in developing new probes to study the relation between structure and function. In normal biochemical literature, structure-function relationship usually denotes the idea that if you make a change to the primary structure of a protein, like sequence of the amino acids, then you change the function. But the way we look at this question is a little bit different here. So imagine having a can opener, and if you have never seen a can opener before, then you may not be able to guess what the function is just by looking at the structure. So that's the structure, but its function is to open a can. And the best way to learn how the can opener functions is to watch someone use it in real time. And as you know, the can opener has to change its shape repeatedly to carry out the function. And this is also true for many of the biological molecules, such as proteins and DNA and RNA molecules. We have wonderful biophysical technologies, such as X-ray crystallography, electron microscopy, and NMR, and so on, to determine the function even at the atomic resolution. We also have wonderful biochemical tools to measure their functions. But really, still a challenging goal is to measure the changes in structures while they are functioning. And this is what we are going after. So we have a technology that we call a single molecule FRET. If you want to measure shape changes or conformational changes of a molecule, you can put two different dye molecules, and green and red, on the known sides of the molecule so that you can distinguish between the closed and open conformations. In the open conformation, if you excite the green dye, then you get mainly green emission. But in the closed conformation, energy can be transferred from green to the red so that you get photons coming out from the red molecule. So by measuring the relative intensities of the two colors, you can deduce the shape of the molecule. Now, if you want to measure how quickly the butterfly flaps its wings in ensemble, you must synchronize the reaction by starting from one conformation. You can convince the molecules, the butterflies, to sit on the leaf in one conformation, and then you can frighten them off into taking off the leaf, and you perform a relaxation analysis. But oftentimes, what you measure here is not the wing flapping frequency, but how long it takes for the butterflies to take off the leaf. Ideally, you want to look at a single butterfly or single enzyme as a function of time, so you can measure anti-correlated changes in the green and red intensities as a function of time. And this can give you information on the amplitude of the motion and also its time scale. So this technology has been really, really powerful in revealing hidden details about biomolecules. It's being used by hundreds of laboratories around the world. So why study single molecules generally? First, you can detect subpopulations and dynamics in the most direct way. And you can also detect non-synchronizable dynamics. If your molecules can be synchronized, just like in a marching band, you don't have to measure single molecules to study the details of the movements. But imagine trying to do that to college students on campus. It's impossible to synchronize, and you have to then observe single individuals. We can also reveal that nominally identical molecules can be heterogeneous. They can have different personalities and different moods. They can only be revealed by looking at single molecules. Finally, when you perform single molecule measurement, you can actually make measurements of multiple observables from the same molecule, make correlations between them. And this is actually a topic of today's talk. There's another powerful technology called optical trap. You can call it chopsticks made of light. You can grab a single particle using a focused laser beam and apply very small forces down to single piconewtons of force. And a piconewton of force is about the weight of a single E. coli bacterium. And it's a very small amount of force because if you have your iPhone, gravitational force on that iPhone is about a Newton. So it is 10 to 12 times smaller than the weight of your phone. If you can grab a single particle and apply a force this way, you can then play a tug of war with an enzyme sitting on a glass surface through a DNA molecule. And this method can give you information on the force being produced by a molecule. And also, you can measure changes in DNA lengths down to an angstrom or single base pair resolution to follow the activities of these molecular machines with the ultimate resolution of single base pair incorporation and so on. So my lab and many others shown on the bottom of the slide have made efforts to combine these two different technologies. One is a single molecule fluorescence, the other is optical trap, so that you can measure, for example, conformational changes of a molecule using a FRET, but as a function of applied force. And the force here actually mimics the physiological conditions because these molecules are in a crowded environment, bumping and tugging each other. And so the measurements performed under tension can be much more relevant to what's really going on inside a cell. If you want, optical trap measurements allow you to measure things mechanically with your hands, but with your eyes closed. Whereas in single molecule FRET, you can use your eyes to make passive observations, but with your hands tied in the back. And by combining the two into one instrument, you can sample the best of both possibilities and reveal new things that you couldn't see before. Okay, let's get into the topic of today's presentation, DNA and the nucleosome. This is a famous paper published more than 60 years ago by Watson and Crick to propose double helical structure of this iconic molecule DNA. And this paper ended with this paragraph saying that it has not escaped and noticed that the pairing mechanism suggests a possible copying mechanism for the genetic material. And then later on, they published another paper proposing how A's and G's can pair with T's and G's can pair with C to form base pairing as the explicit mechanism of genetic material copying, and the rest became history. In our own body, in every cell of our body, you have about 2 meters of DNA. And this long DNA has to be somehow packaged inside a chromosome, shown at the bottom, and inside a cell nucleus. And this happens through multiple stages. At the most elementary stage, DNA is bundled into a structure called the nucleosome, shown in this movie. To look at this structure in slight more details, Here, about 140 some base pairs of DNA is wrapped around a protein core, almost 2 turns, 1.7 turns. So I have seen many protein structures in my career, but this is still my favorite because it's so beautiful. And you just look at it and you know immediately what it means. This is how DNA is packaged inside the cell. The nucleosome has to satisfy 2 conflicting requirements. First, it has to be stable to serve as a compaction unit of our genome in the nucleus. It also has to be dynamic to allow gene expression. Proteins have to come and bind to the DNA to make messenger RNA molecules, but if the DNA is still bound to the protein core, you cannot do that. So my student Tui, shown here, decided to develop single molecule assays to measure the dynamics of a single nucleosome under force, under tension, at the level that is expected to occur all the time inside a living cell. Previous studies done by other labs, shown at the bottom, using the optical trap technology, where they pulled on the ends of DNA in a chromatin, multiple nucleosomes of a single nucleosome, led to the following model, where nucleosome is unraveled under tension in multiple stages. At low forces, outer turn of the DNA in a nucleosome unravels. And as you increase the force to a higher value, then inner turn unravels. And finally, at even higher forces, the protein core is ejected from the DNA. Because we have an instrument that combines single molecule fluorescence, FRET, with optical trap, we decided to test our ability to examine the same process in a different way. So in the previous studies, people actually used the end-to-end distance measurement of the DNA tether to conclude that nucleosome unravels in multiple stages, as shown in a symmetric manner. We decided to put red and green dye molecules to the DNA on the nucleosome, as shown, so that initially, in the absence of any tension, the FRET should be high because of small separation between the 2 dye molecules, high proximity. So you get mainly emission in the red channel of your detectors. But as soon as you unravel the outer turn of a nucleosome at low force, then FRET should go down quite dramatically. And depending on the exact configuration of your measurement, you may see additional FRET decrease upon inner turn unraveling. So that was the idea. This is the actual DNA that we used. We picked a sequence called 601. It's discovered by late John Whittam. This is a sequence discovered from in vitro selection. It's an artificial sequence that is known to position the nucleosome in a precise location on the DNA down to a single base pair precision. Then we can put the green dye near the middle of the DNA. This is called the diode axis, representing the symmetry axis of the nucleosome DNA and nucleosome. And you can put the red dye toward the end of the DNA, so that in the 3D structure shown here, 2 dye molecules are very close to each other, sitting at the entry points of the nucleosome here. So this is a cartoon version of that. So you should get high FRET because of high proximity at the beginning. But as soon as you unravel the beginning part of the nucleosomal DNA, then you should see a major reduction in FRET. And we call this end diode labeling scheme and call it ED1 because we are labeling the diode and end positions of the DNA. We can use a different labeling scheme shown here that we call internal labeling. We move the dyes to different locations so that now the dyes show up at the bottom at the nucleosome. And they are again very close to each other as shown in this cartoon. But you can imagine that you need to unravel more than 20 bases base pairs of DNA before you expect to see a decrease in FRET because you are labeling the internal locations of the nucleosome. So this should be sensitive to the inner turn unraveling that is expected to occur under high tension. Then we can tether the DNA molecule to a surface using a biotin. This is a well-known molecular glue. And then you can pull the other end of the DNA using optical trap through a long DNA linker and measure FRET at the same time. So here I'm showing you the actual data. We have this ED1 and internal labeling probes. So one for outer turn probes and the other is inner turn probes. And I'm showing you FRET as a function of force that we measure from many, many single molecules of the same kind. So you see multiple lines here because these are from different single molecules. For the outer turn probe, ED1, we find that FRET starts at high values at the beginning, at low forces. But as you increase the force to about 3 piconewtons of force, which is a level that is low enough that you should expect to see this level of force all the time inside a living cell, then FRET drops to the lowest value. So major unraveling occurs at low forces, as you expect based on prior studies. Now, if you look at the inner turn probe, FRET starts high because, again, dyes are very close to each other. But the FRET decrease does not occur until you go to much higher forces, let's say 15 piconewton. So that was great because this matches exactly what we are hoping to see, matching the prediction by other studies. Outer turn comes off at low forces, inner turn comes off at higher forces. We are very happy that you could do these measurements, and we should have published that paper at that moment. But we made a mistake of doing one more experiment, and that led to actually a 3-year saga to understand what is really going on, but I'll tell you later that we have a happy ending here. So my students said that, well, if our model is correct, then if you move the probes to the other end or other entry point of the nucleosome, we can call that ED2, then that should also unravel at low forces, as was seen for ED1 over there. But when she performed the experiment, it turned out that ED2 construct actually didn't come down in FRET until you went to even higher forces than the middle one here. So that was really surprising. And if this is true, what it means is that instead of unraveling symmetrically from both ends, unraveling is asymmetric. And it may even be directional, starting from the weak side, propagating toward the middle, and finally to the strong side. Okay, so that was surprising. So we had to do a lot of experiments to make sure that this is not, an artifact of the experimental configurations. First, we noted that in our experiment, the ED2 side, that is a stronger side over there, and is tethered to the surface of a cover slip through a short DNA linker, whereas the weak side, ED1, is tethered to much longer DNA and to the optical trap. So there was actually asymmetry in the configuration. So we thought this may be the issue, and this may be the reason why we see this strong asymmetry. So we did a control experiment where we swap the experimental configurations. In this case, we put a short tether to a surface on the weak side, and then long tether to the strong side. And then when you perform the measurement, nothing changed. What was weak was still weak, and what was strong was still strong. So it's not the surface tethering and asymmetry in configuration that is causing this apparent asymmetry. Now, if this is true, we thought, well, let's say this ED1 is indeed strong, and ED2 is indeed, ED1 is indeed weak, and ED2 is indeed strong. Then if you move the probes a little deeper from the weak side, and we call this ED 1.5, that side should still be weak, maybe slightly stronger. And if you move the probes from the strong side into the nucleosome, we call that 2.5, then that side should still be strong. And indeed, that was the case. So here is a FRET as a function of force. ED1, the original weak side shown in black, unravels at very low forces. Go a little bit deeper, 1.5, shown in red. It unravels still at low forces, but slightly higher. And if you look at 2.5 going a little bit into the strong side, that is still strong, shown in the blue curve. So we began to feel that this must be real, and it's really caused by the intrinsic asymmetry of the nucleosome. But in the end, clinched this idea was the additional measurement that my student did. So she decided to add even more configurations to go around the nucleosome. So I have already shown you 5 different sets of data, 2 on the weak side and then 2 on the strong side, and this is the internal one. She added 2 more data points on the weak side. They still remained weak, and the 2 other data points on the strong side, and they still remain strong, really showing very clearly that indeed nucleosome is asymmetrically unraveled if you apply tension to a molecule. So if you want, instead of unraveling like that, shown here, we may have a system where we unraveling occurs in this manner, like that. Okay. So the next question is, why is it asymmetric? And another follow-up question would be, is it important biologically? So we then looked at that 601 sequence in more details. So this has 147 base pairs, and we say that there's a strong side and there's a weak side on the DNA. And it turns out if you look at the nucleosome structure itself, the protein part in the middle is completely symmetric. There's no asymmetry there. So it must be the asymmetry in the DNA sequence itself, which in fact is not symmetric. So the idea we had is shown here. We thought perhaps the strong half is strong mechanically because DNA sequence makes it more flexible. Why? In a nucleosome, the DNA is sharply bent around the protein core. And this sharply bent conformation is not natural for the DNA. So it wants to become straight. Right? But if the sequence makes the DNA more bendable and more flexible, then this sharply bent conformation is better tolerated even under tension. And that can be responsible for stronger mechanical stability of one sequence over another. So that was our prediction. But how do you test this prediction? Well, we need to be able to measure flexibility of a DNA of very, very short lengths, about half the length of a nucleosomal DNA, 70 to 80 base pairs. We are lucky because my former student, Reza Vafabakhsh, had developed an assay that can just do that we call this single molecule looping assay. And here, we start with short DNA, about 100 base pairs in length, immobilized to a surface using biotin-avidin interaction. And then we have sticky ends on the both ends of the DNA that are complementary to each other, so they can base pair against each other. If you start with a condition, low concentration of sodium chloride, for example, that prefers this linear, unlooped state of the DNA, and then rapidly change the solution condition to go to high sodium chloride concentrations that favors this looped conformation, then you can measure using FRET, for example, how quickly the loop population develops as a function of time. Okay, so this is actually data, fraction of loop molecules versus time, and shows that for this particular sequence, looping takes about 15 or 20 minutes. Now, the idea here is that if the sequence makes the DNA more flexible, then looping will occur faster. Then you can use the speed of looping or rate of looping or looping rate as a measure of flexibility. Okay? So we decided to measure looping of the sequences found inside a nucleosome. I told you we have two halves, strong and weak halves. So on the bottom, I have a looped fraction as a function of time. For the red curve, that's coming from the strong side, that took about 27 minutes. Whereas on the weak side, the green curve took more than 3 hours. So indeed, the data suggests that strong half of the nucleosomal DNA is actually much more flexible, as we predicted. By how much? About 7 times, according to our measure of flexibility. So we are very happy that our prediction was actually supported by the data, but this also could be a coincidence. So we decided to look at this in more details. Let me just tell you what we did. So we then divided the DNA into 4 segments, 4 quarters, and then first decided to change the sequences of the DNA so that we can swap the outer quarters of the DNA sequences. And we performed the single molecule looping experiment and the optical trap experiments. And we found that nothing changed. What used to be strong was still strong, and what used to be more flexible was still more flexible, and so on. So it's not the outer quarters of the DNA that are causing the asymmetry. If you instead swap the inner quarters, as shown in the color scheme here, while keeping the outer quarters the same as the original DNA, then everything swapped. Now, what used to be strong became weak, and what used to be more flexible became stiffer. So we now believe that it is the sequence of the inner quarters of the nucleosomal DNA, and the resulting flexibility is a determining is a determining factor for the mechanical stability of the nucleosome and the asymmetry that we observe. This paper was published in Cell in 2015, and I told you there would be a happy ending here. So she was very happy. Then she went on to study the effects of not only the DNA sequence, but also of DNA chemical modifications. As you may know, cytosine C in the DNA, methylation is an important modification that can determine gene expression and epigenetic memory. When she performed the measurement of DNA looping on highly methylated DNA, she found that that makes the DNA less flexible, and then also found that nucleosome, as a result, becomes weaker mechanically. And then when she examined additional cytosine modifications shown here that are important intermediates while you're trying to remove the methyl group from the cytosine, this makes the DNA much more flexible, even with a single modification, and nucleosome much, much stronger. So even here, we find that whenever you make changes to the DNA to make DNA more flexible, nucleosome becomes stronger, and vice versa. This correlation holds every time you make a change to the DNA, and so much so that if we find an exception in the future, it'll be an interesting exception. So the outlook here is the following. We now want to use high-throughput genome-wide technology, such as sequencing, so we can measure flexibility of the DNA across the entire genome. And because of the correlation between the flexibility and nucleosome stability, we can then use that information to predict how stable individual nucleosomes are inside the cell. And then used this as a proxy to make a connection between the physical properties of DNA and the gene expression and other processes involving chromatin. And we are hoping to discover the signatures of the flexibility, that are imprinted in our own genome throughout the evolution process, if indeed DNA flexibility is important for biological functions. Thank you very much. This is just a slide to advertise the third part of this 3 talks series, where we will now use even more advanced instrument that combines ultra-high resolution optical tweezers that can measure DNA processes down to a single base pair resolution with a single molecule FRET to measure conformational changes of a single helicase enzyme and its activity all at the same time and make important correlations and discoveries. Many of the lab members have contributed to these studies, especially Chui Nuo, who did bulk of the research. We also acknowledge collaborators in the lab of Aksmentiev and also in the lab of Chuan Hei in the University of Illinois, Urbana-Champaign, and University of Chicago. Thank you.